I welcome all of you for the technical session now. Now I humbly request our faculty, Dr. S. Palparumal, to chair the session now.
I have to allow it first.
With infectious disease without vaccine, there is no safety in numbers. With infectious disease without vaccine, there is no safety in numbers. Quotes by Seth Berkeley. Good morning, everyone. I extend a warm welcome to all International Conference on Emerging and Re-Emerging Infectious Disease 2023. I am greatly honored to introduce our chief guest, Dr.Vaithilingaraja Arumugasami, Professor, Molecular and Medical Pharmacology, Member CTSI, University of California, Los Angeles, USA. In 2004 to 2008, he worked as a postdoctoral fellow in University of California, and 2008 to 2010 he serves as assistant researcher in field of molecular and medical pharmacology. In 2017 to 2019 worked as an assistant professor in University of California, Los Angeles DeWitt Caffeine School of Medicine. His research focused on understanding the pathogenic mechanism of Flavidae virus family, hepatitis C virus, Zika virus, and dengue virus. He is mainly focused on to develop a potent recombinant vaccine to prevent the Zika virus infections. He also developed a reserve genetic system to engineering attenuation mutation in Zika viral genome and animal models to test the safety and immunological profile of these engineered vaccine candidates. In addition, he investigating the differing impact of sexual and mosquito vector mode of transmission in the infected adults of fertile outcome, and also evaluating the genetic and molecular basis of specific cellular perturbations allow for understanding the basic biology of eye development and identifying novel therapeutic interventions. I feel very happy to introduce the eminent person to this ICERID 2023. Now I invite his enlightened the participants with his word. Welcome you, sir.
I thank Dr. Bala for the invitation, and also Dr. Joseph Ignatius Irudayam for having me here. It's a great pleasure to give this talk to VHNSNC Kumar and Nadar College students and faculty. I'm a faculty at Department of Molecular and Medical Pharmacology at University of California, Los Angeles. Some of the slides I plan to present, the students Nate and Aishi have already presented, so I will skip those slides. But I will try to also keep the talk shorter because Dr. Arun has to present next. Nate, next, please. The outline is, I just want to show, the life, early life I took, and my career trajectory, because we have master students and PhD students. This may be, useful as they plan for their next step in career. And also, I'll touch base with the Zika virus project we've been doing. But Nate, he already presented the hippo signaling pathway, so I probably won't go too deeper into there. And also, we are one of the first few labs starting to work on SARS-CoV-2 virus. So I will share some of the story we've been to. And also our current antiviral research efforts on multiple pandemic potential viruses. So Nate, next, okay, next, Nate. Yeah, I'm from Kadayam. This is a small town near Kuttalam. That's where I started my I was born there. Then next. This slide I used for another US talk where I tried to show them many of the South Indians also leaders in key institutions. Next, please. So I went to Madras Veterinary College. That's where I got my degree. So I'm a veterinary doctor. From there, I went to National Institute of Indian Veterinary Research Institute in '97 to 2000. It was a leap from Tamil Nadu to going to Delhi. There, I studied my master's of pathology for 2 years. The the institution was built very early on during colonial era, but the place is next to Himalayas, so you could actually see those main Himalayan peaks from the hostel. This is at the Mukteshwar. That's where all the veterinary important viruses they do culture them, and they many of the vaccine projects were ongoing at that time. So from Indian Veterinary Research Institute, I moved back to United States in around 2000, where University of Arkansas, where I did my PhD. It's a Fayetteville. It's a small town, college town. Then I took a leap to UCLA in 2003 or 2004. So I've been in Los Angeles for last close to 20 years, or you can say. And I'm very fortunate to have great students and also colleagues, mentors here. So next, please. So our lab deals with a lot of high consequence RNA viruses. For example, Zika virus can cause brain damage to developing fetus. SARS-CoV-2, from the current pandemic. There's another virus, hantavirus, which actually the rodents, the mouse or wild rodents can spread this virus through their urine. This is another kind of a ticking time bomb. I will explain some in the later slides. And also chikungunya virus. Chikungunya virus, you may have heard about that. In 2007, there was a huge epidemic around Indian continents and also, I think, Pacific Indian continents. It caused a lot of morbidity. Not the mortality, but people were sick with this virus. So we use technology like RNA sequencing, proteomics, metabolomics, many of the omics, lipidomics, to study these pathogens. Mainly, we focus on understanding the host pathogen interactions, the virus interacting with hyposignaling pathway, innate immune pathway, and also how the virus causing cell injury and death. We do use many model systems because viruses, you as a microbiology students, you cannot grow the virus in petri dishes. You do need living cells. So we use cells, organoids, mouse models to study these pathogens. And mainly we focus on getting the drug developed against these viruses. Next, please. One thing, before this pandemic, many times I felt microbiology field was not recognized as it should be. But now there is, because of the pandemic, there is a lot of requirement for microbiologists in the field. So if you are thinking about career in virology or bacteriology, also even mycology or studying fungus, I think this is the time. This is a great time to be in microbiology. So if you look at this timeline, 1960s, we already know coronaviruses are there. They cause kind of human seasonal flu-like disease. But as we go through 2002, we got the first SARS-CoV, and also 2012 and 14 MERS-CoV, now the SARS-CoV-2. If you look at the similarly, next one. You can keep looking at other viruses like chikungunya virus, Zika virus, the last 2013 to 20, you see outbreaks from all these viruses. So if you look at if you ask a microbiologist or infectious disease specialist, uh, these disease outbreaks are commonly happening, but people are not aware of it. Only now the pandemic created that awareness. So we need to study these viruses. Next, please. So now there is a priority, at least in throughout the world, specifically in United States. There is a clear focus on studying coronaviruses, paramyxoviruses, flaviviruses, tocoviruses, filoviruses, and enteroviruses. Viruses typically use our weakness for breathing. Can you go next? Okay, so yes. So breathing, if you stop breathing for 3 minutes, you would die. Same way, if you don't eat for 3 to 21 days, you die. Same way. So the virus actually using our weaknesses for to spread. The respiratory pathogens are extremely successful in that way, as we learned from the SARS-CoV-2, and also the mosquito-borne virus like dengue virus, Zika virus, and also West Nile virus here can cause over billions of infections per year. If you look at why we are getting viral infection, look at when you are going deeper into the jungles in Kerala or Assam, you're going to have exposed to viruses. They are native to those jungles. They can transfer to you. For example, Nipah virus. It's a very fatal brain damage causing virus. You go deeper in the jungle, you're going to get it. Same way in the SARS-CoV-2 outbreak, the COVID-19 pandemic, many of the big cities actually hit harder. Similarly, now you see poliovirus start coming back, and a lot of enteroviruses, and also mosquito-borne viruses, so mainly because of the unhygienic conditions. And also one thing more peculiar for this pandemic, people with diabetes or hypertension or obesity actually had a higher mortality rate, if you understand. So as the population growing and people are having more diabetes or hypertension, comorbidity conditions, we're going to see more of these viruses affected. Next, please. So I think I can skip this hippopathway section. Ned, can you just keep? Yes. Okay, yeah. So one of the projects we just completed and published is the Zika vaccine. So this is when the Zika virus actually infecting a pregnant mother, the pregnant mother feels very mild fever or rashes. There's not much issue with the adults, but the virus can actually pass through the placenta, reaches the developing baby, and can damage the brain or eye. So we essentially focus on developing a safe and effective vaccine. So this study we published 2 months back. It is a well-funded study. So where we can actually split the virus into two components using genetic engineering and produce the vaccine particles in cell culture. Then we tested their safety and efficacy in the animal system. Next, please. So what you see, at least you can focus on the fetuses. These are the mouse babies, and the vaccinated, the pups or the fetus coming from vaccinated mom, they are very healthy. You can see in the bottom cluster. But on the top, unvaccinated pups or the fetuses, they usually very small. You could see there will be dead and decomposed pups in the uterus. So our vaccine very effective, and this study recently published. Next, please. Dr. Arun Ramaya, so when this pandemic was happening, or it's not even a pandemic, they said in January 2020, mid-January, they were talking about 400 people infected in a city, Wuhan, in China, with 8 people died. So for me as a virologist, it is a red flag. Recently with Zika virus, there is no adult died at that time. But with this new virus, there are 8 people died in 400 infection. So we thought this is something very unusual virus. So we started studying this virus very early on. Dr. Ramaya will present more on this evolution study and the current trend in the transmission or the variants. But next one. So in March 2020, nobody cared in USA. There are one case identified in Washington, but we want to study. This virus is new, unique, and we immediately got this virus from CDC, American CDC, and we started growing them. So wherever you see red color, that's where they are the infected cells. We used a viral spike or nucleocapsid antibody. And also the panel C, we tested number of drugs. You could see hydroxychloroquine, interferon beta, those drugs are working. So, and also panel A, if you see Vero E6 cells, in the 2 days post-infection, SARS-CoV-2 infected cells, they look very unhealthy, and we call it cytopathic effect, CPE. So for this study, next please. Gus, I'm on the top. Gus in the middle. Gus is kind of a hero in this effect. He was almost 24/7 worked in the lab, did several outstanding studies. Some of them I will share here. And also Barbara Dillon, director of BSL-3. She was running the facility nonstop. So for this virus, we do need to use a really high level of high containment facility because the virus at that time, they don't want this virus to escape from the lab to go outside. There is a theory this virus may have come from a lab, but we don't know for sure. But you don't want the virus escaping from the lab. So it's all done in a very high secure facility. Next, please. Nate presented this slide. This is a lung cell culture. It's a very advanced culture system. Maybe in the world, maybe 10 labs can do this. You need a human tracheal cells to establish this culture and do the infection. These are the first waves of papers we published, cells, stem cell, maybe it's impact factor 35 or 50 something, very high level of impact factor. Then also were highly cited. These papers are highly cited. If you get a chance, you can PubMed it or Google it. You'll understand the importance of these work. Next, please. I think this may not work. The video. Can you try? If not working, then we can move to next slide. Yeah. Yeah, next slide, please. Yeah, this is another important study where we could able to grow virus in the human lung cells. The green you see are the SARS-CoV-2, the red are the human lung cell marker. Next, please. And also, we are the first group to show the SARS-CoV-2 can directly infect human heart cells. This is one of the highly cited paper in the field, where at that time we know the people with heart problem or hypertension, they are susceptible to this virus, but we showed direct evidence the virus indeed can damage the heart cells. Next, please. You think the video would play, or we can skip if not playing? Okay. Anyway, this is the kind of a organoid system. It's a human heart cells beating. Um, when you add, or these cells are infected with SARS-CoV-2, the heart cannot function, so the cells stop beating. So we measured how many heartbeats, and with the infection, there is no heartbeat. This was published very early on. Next, please. Nate, next. Yeah, these are the papers we published during the heat of the COVID-19. Many of the cell culture systems, we were the first one to publish. The virus can affect the neurons as well, brain cells as well. Next, please. So this is another critical slide, or the concept for microbiology students. So when the companies were developing antivirals, or the tablets or drugs for HIV or hepatitis C virus, these viruses are very slow causing, or slow viruses. They cause disease in years, maybe 20 years or 30 years or 10 years. So you have enough time to stop this virus, and treat, and the patient will be better. But in acute viruses like SARS-CoV-2, next one, SARS-CoV-2 or dengue, you have a very mild symptom. Then some people progress into a severe or moderate to severe symptom. So it's very difficult to spot where we can start the treatment. So essentially, when you start the treatment in later on at the severe stage with the antivirals, it won't work, because by the time the virus damage the body is responding with inflammation. So at that time, you need to treat the host as well. So you need a kind of a combination of therapy. Next, please. So we set out to do a drug screen. This was done very early on during the pandemic, March 2020. We quickly did the drug screen. For this study, we used Vero cells with SARS-CoV-2, and also about 430 kinase inhibitors. And we identified a drug compound, berzosertib. This is kind of in that screen. This was very top hit, and we actually secured a military funding to study this drug compound in detail. Next, please. So here, this slide shows our drug screening, the drug hits, and wherever you see less red means the drug is working. So that is a kind of a take-home message. And based on the number of drug hits, we did a network analysis. Next, please. So this is the network analysis. Most of the compounds were actually clustering around mTOR-AKT pathway. So that was another study we published in Nature Communications, where we showed the SARS-CoV-2 actually affect the mTOR signaling pathway. It's an energy pathway. Next, please. So this is also a video. It may not work, but the drug we developed or identified can actually cause the SARS-CoV-2. It inhibits the SARS-CoV-2 and also rescue the heart cell beating. Next, please. So this is our current effort, where we also study MERS-CoV-2, not a COVID-2, MERS-CoV. This virus fatality rate is 40%. Means if 100 people affected, 40% of the people, or 40 people, will die. So this is a very high virulent pathogen, and we can able to actually use drug to reduce the replication in callus cells. This is our ongoing study. Next, please. And also, We do study other viruses spread by mosquitoes. They are chikungunya virus. We have West Nile virus, uh, Zika virus. We actually use uh, uh, eye organoid or brain organoid. So this study was actually mainly being driven by initially Gus, but then moved on by Dr. Joseph. He's here in this audience. This is one of the most challenging project because you have to grow the stem cells, then you have to add expensive cocktails of cytokines to make the stem cells to become a brain cells or eye cells. These are a very highly cutting edge technology. This may take 3 to 6 months of study to get some data. So this is very, very viral infection. Next, please. And also, we actually did a lot of screen for these chikungunya, West Nile, and Zika virus, and we show in the animal model that drug treatment actually can reduce the swelling in the leg. Chikungunya, this virus, my mom got affected with this virus in 2007. Still, she has pain in her hands. Same way, if you talk to your relatives, at least some of them still having the pain from infection 15 years back. So this is a very chronic disease, and we have able to develop a drug which can actually reduce that inflammation. So this study is actually currently being in revised. Next, please. And also, this is another virus, hantavirus. This virus actually described in the World War I as well as World War II, where the soldiers in the trenches, they get kidney failure. But now they have this virus actually evolved, and the virus actually now evolved in the Alps mountain in the South America region, actually can spread from human to human. It used to be spread by rodents to human. The human cannot spread this virus. So currently our major effort is on this virus, identifying drug compounds and understanding this virus better. So this is one thing microbiology students can write it down. This could be one of the viruses can cause next pandemic. That can be pretty devastating. It has this virus can spread through respiratory route, and in cell culture, this is very, very. It's very similar to MERS-CoV or SARS-CoV-2 in terms of killing the cells. So this virus need to be watched out. And also, the virus can be used as a bioweapon, especially the Rift Valley fever virus. So the virus been around in the African continent, but mainly in the cattle and goats. But the virus can spread to human being. That can be used as a weapon. So we are studying all these viruses and developing drug compounds against them. Next, please. I think I'm almost in the last 1 or 2 slides. So during the COVID-19 outbreak, we are 1 or 2 labs in the west coast of United States doing the SARS-CoV-2 work. So that time we worked with many of the people's high-level scientists, and we had a great success. And some of the collaboration is still ongoing. And also a couple of postdoc fellows from this group actually become a faculty. So that's the kind of high point. Next, please. And we got many of the press releases, worldwide news coverage for many of our discoveries. Recently for Zika virus and also SARS-CoV-2 antivirals. Next, please. So it is not possible without a great team of people working hard on developing or progressing on. So Gus is one of the man to go, and is a great scientist. And we also have other number of people. So Arjit Vijay is here. He's a bioinformatician. Now he's the one driving the Antiviral project. Zubu, he just joined our lab. He is actually taking over the hippo signaling pathway. Nate and Ayushi, they just presented. And Ramya is here. Ramya is another undergraduate student supporting Nate. And also I suggested Joseph Irudayam. So it's a lot of hard work, and also we do have fun. I don't know, we have next slide. So that's my family. I have two daughters, but now they are my height. They are taller. And also, this is Einstein. I just at least try to be. Next one. So this is when I was 20 years back, young, I used to play in the USA Open. It was a fun time. But now mainly we doing, next one. Just fun volleyball with friends. So, and also we in Los Angeles, we have great mountains to hike. So whenever you get a free time, you climb in a higher level mountains, and it's also challenging. So this is one way we can wind up. So if any of you are interested in coming to USA, or you have an ambition to be a great scientist, UCLA is one of the best place to be. It's close to the beach, and also you can go to hike 11,000 feet elevation soon. So with that, I will stop. If you have any questions, I will take. You can actually send me the questions. If there's not much time, I'll let Dr. Arun take over from here. Thank you.
Thank you very much, sir, for sharing your precious knowledge as well as your remarkable memories to us today. And thank you very much for your wonderful presentation, sir.